We know that the DNA has specific base sequences. The DNA molecule has bases. Bases, you know, has bases. And in the DNA molecule, a specific base sequence which determines the character of an organism. The specific base sequence which determines the character of an organism is called gene. The base sequence is named as a gene. A specific region of the DNA molecule which is responsible to create a character in the organism. Which is responsible to create a character in the organism is called the gene. What are the characters we have? We have characters, hair color, eye color, skin color, height in the body, and the appearance. They're all are characters. So these characters are determined by our genes. So what is a gene? Gene is a specific region present in the DNA molecule which is responsible to create a character in the organism. So then this sequence makes one character, and another sequence may create another character. Another sequence may create another character. The DNA molecule determines the characteristics of living organism. That is why the DNA is identified as the genetic material of the living organism. It is the genetic material of living organism. Why? Our characteristics, the information for our characteristics are stored in the DNA molecule as what? As specific base sequences. Now, how this base sequence determine our character? Is this DNA is making the character, or like, so there should be a method for that. Yes, by explaining about the gene, yes, we explained that the gene is a particular base sequence, a region of the DNA molecule, which determines the character of a living organism. So my question, how it create the character? What is the mechanism of creating the character? That is what we are going to discuss as our next lesson. So first we have to consider about the character. Characters means not like our personality and the humanity, not such characters. Right? Here we are talking about our physical characteristics. The body and the chemicals inside the body, and reactions inside the body, the arrangement of the molecules inside the body. So those are our characteristics. So those are the characters we are talking about. Remember, these characters basically are made up of protein molecules. Our characters are made up of protein molecules. Example, our hair, protein, keratin. And the muscles, protein, actin, myosin, protein. Nails, proteins. Bones have protein, ossein. And cartilage has proteins. Blood contains proteins, plasma proteins. Enzymes, proteins. Antibodies, proteins. Okay. All the structures in this body are protein structures. So therefore, determining a character means making a protein. Making a protein. Now DNA is responsible in making a protein. That is what we call DNA determines our characteristics. So the DNA gives information to synthesize a specific protein. What is a protein then? You have the idea about protein. What is the protein? Protein means a specific amino acid sequence. Protein is a specific amino acid sequence. Amino acids. Amino acids are arranged according to a pattern and create a specific protein. According to the sequence, it determines the structure of the protein. primary, secondary, tertiary, or quaternary. According to this amino acid sequence, I'm talking about proteins now, not DNA. Now, according to this amino acid sequence, it determines the structure of the protein. Structure of the protein means our character. Because our character is created according to the structure of the protein. When the amino acids are arranged as a specific sequence, it may create keratin protein. It can be identified as keratin protein. In another amino acid sequence can be identified elastin protein. Another sequence can be identified hemoglobin. Another one can be identified as collagen. So why we have varieties of proteins there? Because of the differences between the amino acid sequences. The arrangement of amino acid sequence determine the structure of the protein. Arrangement of the structure of that protein determine the character of the living organism. Then, for example, our hair. Our hair has specific structure. Hair is made up of keratin proteins. Then keratin protein has a specific amino acid sequence. When it creates that amino acid sequence only, it creates the keratin protein. If the amino acids are not arranged according to that pattern, it's not keratin. Then it creates the keratin protein. Then our hair can be developed. Like that. Then the amino acid sequence is the one which determines the structure of the protein and then which determines the character of the living organism. Who gives the information to this amino acid sequence? Someone should give information to arrange the amino acid according to a pattern. When the amino acids are arranged according to that pattern, it creates the protein. So then who orders, who give the order to arrange those amino acids according to that pattern? DNA. According to the base sequence in the DNA, amino acids are arranged. The pattern of amino acid, the sequence of amino acid is determined by the sequence of bases in the DNA molecule. So let's see what is how the mechanism that creates there. Let's see the mechanism. Then first get the concept properly. Get the concept properly. If you have the concept properly, then it's easy to identify the mechanism. Okay. Now, the DNA has specific base sequence. Adenine, cytosine, guanine, thymine, adenine, cytosine, guanine, adenine. Like there's a base sequence in the DNA. This base sequence determines the order of amino acids. This order is arranged according to this base sequence. When the amino acids are arranged according to this pattern, it has a specific structure. The sequence of amino acids determines the structure of the protein. Structure of the protein means the character of the organism. Then our character is created by this polypeptide chain or amino acid sequence using the information provided by the base sequence in the DNA molecule. This is what we say that our characters are determined by our genes. The base sequence means the gene. So this process is called the protein synthesis process. Protein synthesis. Have we cleared the concept of this one?
Yes,
sir.
Right. Now, so what is this protein synthesis process? First, we need to take the DNA molecule. DNA molecule. Then think that now the DNA, one of the gene in the DNA, now is going to synthesize the protein. Think. Now, then the gene. So this is the region of the gene. The specific base sequence is here. Now, the specific base sequence will provide the information to synthesize the amino acid sequence. This process happens in two ways, two steps. This process happens in two steps. That means protein synthesis process happens in two steps. First step is called transcription. Transcription. What happens during this transcription process? The base sequence in this DNA molecule is copied into an mRNA molecule. The base sequence present in this gene, the base sequence present in this gene is copied into an mRNA molecule. mRNA means single-stranded molecule, RNA, messenger RNA. Okay, so I will explain the mechanism, but identify the concept step by step here. Okay, first what happened? The information present in this gene is copied into a mRNA molecule. Right? It's copied into an mRNA molecule. The process is called transcription. Then, next step is second step. This is first step. Synthesize an mRNA molecule. Next step is translation. Translation means, according to this base sequence in the mRNA molecule, which is given by the gene, amino acid sequence is synthesized by the ribosomes. Ribosomes synthesize amino acid sequence according to the base pattern present in this mRNA molecule. That is what happened during the translation process. In transcription process, it synthesizes an mRNA molecule. In translation process, it synthesizes the amino acid sequence, polypeptide chain. These are the two steps of protein synthesis: transcription, translation. Simple concept should be identified first. Still, I didn't explain the mechanism. Gene sequence, the bases in the gene, or base sequence in this gene is copied into an mRNA molecule during the transcription process. Remember, this process occurs in the nucleus. This happens inside the nucleus. Transcription process happens inside the nucleus. Why? DNA is located in the nucleus. This copying process, this happens in the nucleus. The mRNA molecule synthesized inside the nucleus. And this mRNA then coming out from the nucleus through the nuclear pores to the cytoplasm. Then this translation process takes place in the cytoplasm, because ribosomes are available in the cytoplasm or the endoplasmic reticulum. This is the process of protein sequence. Then let's see the mechanism of transcription process. The mechanism of transcription process. Okay. Now, in the DNA molecule, this is the DNA molecule. The DNA molecule. Think that this is our gene region. Okay, our gene. Now, what happened here? In the nucleus, there is an enzyme called RNA polymerase, not DNA polymerase. RNA polymerase. In the nucleus, there is an enzyme called RNA polymerase. So what happens here? RNA polymerase binds to the starting point of this gene. There are regions called promoter-operated regions. Still, I am not explaining about those regions. I will explain it later. First, identify what's happening here, the general explanation. RNA polymerase binds to one end of the gene. RNA polymerase, not DNA. DNA polymerase used to synthesize DNA. Here, what is the target of transcription? To synthesize mRNA. To synthesize mRNA, it is required RNA polymerase. That's why the RNA polymerase binds to this DNA molecule. Okay. Now, when the RNA polymerase binds to this region, what happened here? RNA polymerase. So that particular region, in the particular region where the RNA polymerase binds to the DNA, is the DNA strand separated. In that particular region only, it creates a loop-like structure. Two strands get separated because of the action of RNA polymerase. It breaks the hydrogen bonds. It breaks the hydrogen bonds and separate the DNA strand in that particular region by the RNA polymerase. Now it's meaning that strands separated, bases are open. Bases open because hydrogen bonds break. Individual bases are now open. There. What happens here? When the bases get open, when the bases are getting open, what happens? The free ribonucleotides will deposit in front of these open bases according to the complementary base pair. RNA ribonucleotides deposit here. Okay. For example, think that this particular DNA strand has adenine, guanine, thymine, cytosine, thymine, adenine, thymine. The separate strand has this base sequence, this particular region. So, what are the ribonucleotide deposits in front of these bases? Adenine. What is the base?
Thymine.
No, in DNA, RNA, there are no thymine. If it is RNA, that should be uracil. In RNA, we can find uracil. Because the ultimate goal here is to synthesize RNA molecule. Therefore, when the RNA polymerase unwind the DNA molecule, the ribonucleotide carries uracil base to the adenine base. Others are not changed. Thymine, cytosine, adenine, cytosine, adenine. This is uracil. When adenine is there, the base is uracil. Like that, what happens? The bases are deposited and it makes phosphodiester bonds between these nucleotides. Phosphodiester bonds are created between the nucleotides. Okay, give me one time. Light take it for disturbance. Understood? Then this RNA polymerase continues to move along the gene. RNA polymerase is moving along the gene. When the RNA polymerase is moving,
sir? Sir?
yeah,
Does the RNA deposit only on one side of the strand?
yes,
Sorry,
yes,
only on one
yes,
strand?
yes, now we do because that what matter that DNA polymerase move from 3' to 5' direction. So RNA polymerase also moving from 3' to 5' direction. So then when it's moving to that direction, only one side of DNA deposit with this ribonucleotides. So that is that only one strand get read. Other one is not reading.
Okay.
Right? The reason is that because that particular enzyme is moving from only to one direction, 3' to 5'. When moving it, it synthesizes the RNA in that strand only. Okay? That is the reason. So only one strand gets synthesized. Then what happened here? The RNA polymerase is moving. Now RNA polymerase is now here. The bases are open here. Then our cutting area is now it's again complementary bases paired and the DNA synthesized. What happened to that early synthesis? This fragment. What happened to that fragment? It's coming out. So as the RNA polymerase is moving along the DNA molecule, the synthesized mRNA is coming out from the molecule. Then two strands of DNA rewind again. They get again the bases get paired. But the place where the RNA polymerase stands, again the strands get unwind and ribonucleotide deposits, mRNA is produced. So when the RNA polymerase is moving to this direction, mRNA is synthesized according to the base sequence present in this DNA molecule. So finally what happened? At the end, now the DNA is there, the gene, what we have selected here, gene is there. DNA RNA polymerase detach and it's leaving. And newly synthesized mRNA is present. mRNA. Newly synthesized mRNA is present. Okay. Now here, the base sequence presented on this DNA is now copied into mRNA molecule. Think that DNA strand think this is the strand which read by the RNA polymerase. It had adenine, cytosine, guanine, thymine, adenine, cytosine, thymine. This is the base sequence. So then what is the base sequence in the mRNA molecule now? Here adenine, it should be uracil, guanine, cytosine, adenine, uracil, guanine, adenine. This is the mRNA sequence. This is the mRNA sequence now. The information copied into the mRNA. Messenger mRNA. Messenger RNA. Now this messenger RNA is synthesized inside the nucleus. Now it will be coming out from the nucleus through the nuclear pores to the cytoplasm for the next step. Is that clear?
Yes, sir.
Transcription process. Navindu?
Yes, sir, clear.
Okay. See the first step of DNA synthesis, transcription process.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
See how the DNA is arranged in the chromosome. This is how it shortens the length of DNA by wrapping surrounding the protein beads, which are called histone. The DNA is wrapping surrounding that histone protein to minimize or to shorten the length, then arrange in the chromosome.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene.
Now RNA polymerase attached to the starting point of the gene. Then, as I explained, it gets unwound. That particular point, the DNA strands are getting unwound.
It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production.
Okay. How the mRNA is synthesized now. So, most important part is the base sequence of the mRNA is complementary to the base sequence presented in the DNA molecule. That's what I said, that DNA information is copied into an mRNA molecule. Now, mRNA is synthesized according to the base sequence in the gene. mRNA is synthesized. The base copied into the gene. So this process occurs in the nucleus and the mRNA comes out there. Next one. Now mRNA molecule is coming out here from the nucleus and now it is in the cytoplasm. Remember, when we analyze this mRNA molecule, the bases in the mRNA are arranged as base triplets. The bases in the mRNA molecule are arranged as base triplets. Each triplet is named as a codon. Codon. Here triplet, another triplet, another triplet, another triplet likewise. Because during the step, I am going to explain the translation process. Because during the translation process, this mRNA molecule is reading by the ribosome as triplets, base triplets. Now ribosome is going to read this mRNA sequence. When the ribosome is reading the mRNA sequence, it is reading as base triplets, 3 by 3. Okay, that is why we said that mRNA is arranged as codons, the bases. For example, Now, AUG, CUA, UUC. Likewise, it is arranged there. So therefore, මම ඒක ඈතින් ඈත අඳින්නනේ. එහෙම නෙමෙයි තියෙන්නේ. ඈතින් ඈත නෙමෙයි තියෙන්නේ. But I will draw like this to understand well. Okay. But there are no gaps. There are no gaps. But for our the explanation for our the lesson to explain this, I will draw separately as base triplets. Codons. Think that this codon is AUG. UAC, CCU, like that codon. Now what happened here? Ribosome binds to the mRNA molecule. Ribosome binds to the mRNA molecule. Okay. Now these ribosomes have specific rooms. There are rooms. I am not going to explain that into that detail. But so here the ribosomes. Now codon is present inside the ribosome. The ribosome binds the mRNA molecule. Then in the cell cytoplasm, there is another RNA molecule which is called tRNA. Transfer RNA. There is another RNA molecule which is called tRNA. Transfer RNA molecule. Okay. So this transfer RNA molecule, the shape of the tRNA is like a hairpin. It's like a hairpin. It's a single-stranded RNA. Single-stranded, but because of the complementary base pairing, it is folded and it creates this hairpin structure. Like this. The complementary bases are paired. Then, මේක දාන්න මේ ස්ට්‍රෑන්ඩ් එක කියලා මේක. මේ ස්ට්‍රෑන්ඩ් එක මේව නැවෙන්න පුළුවන්. මොන මෙයා දෙකට නැමෙනකොට මෙහෙම යනකොට so if they meet complementary bases they make hydrogen bonds ek position එක complementary bases තිබුණොත් දෙපැත්තේ so then if two sides get complementary bases they make hydrogen bonds so then because of that the tRNA creates this hairpin structure but it is a single stranded. Understood? Right. Now, okay. So now the tRNA molecule is the structure of that tRNA molecule. So what is the important part? This tRNA molecule, when you take this tRNA molecule, it has bases, open bases. It has open bases which are not complementary paired. Open bases which are complementary to the bases in these codons. Right? We have tRNA in the cells. We have tRNA in the cells which has triplets of bases which can be complementary to any one of the codon in mRNA. That's the most interesting thing. So we have tRNA in the cell. Those tRNA has bases, triplets. Those base triplet, open triplet, can complementary to one of the codon in any mRNA molecule. This triplet is called anticodon. Anticodon. tRNA has anticodons. tRNA has anticodons which can be complementary to one of the codons in the mRNA molecule. Right? And also, according to the base sequence anticodon, it can be attached to an amino acid. Right? It can be attached to an amino acid. This amino acid is determined by anticodon sequence. According to the anticodon sequence in the tRNA, a specific amino acid is attached. So there may be another tRNA in the cell which has different anticodon and it will attach to another amino acid. Because we have 20 different amino acids in the body to create proteins. So then nearly we may have 20 different tRNAs. We have 20 different amino acids. So then this tRNA, that's why it's going to transfer RNA. Now it is going to transfer these amino acids to the ribosome, which is going to read the mRNA sequence. Okay. Now the process is starting. Think this anticodon has UAC. Then UAC is complementary to AUG. Then what happens? This tRNA is transferred into the ribosome and pair with this and it gets paired with the bases present in the mRNA. Right? Bases paired with that, and also it carries its own amino acid. Its own amino acid is there. Right? And then ribosome is moving along the mRNA molecule. Ribosomes start to move along the mRNA molecule. Okay. So what happens there? Now the ribosome is moving along the RNA molecule. Then another tRNA comes according to the complementary base pairing of the bases, the codon and anticodon, to the ribosome, and it carries another amino acid. This amino acid determined by the anticodon. Then what is the anticodon connect to mRNA is determined by this codon. මේ codon එකට set උනේ specific anticodon එකක්. ඒ anticodon එකෙන් තමයි determine කර දෙන්නේ what is the amino acid taken there. So ultimately we can say that this particular amino acid determined by this codon. මේ codon එකෙන් තමයි determine කරේ amino acid මොකක්ද කියලා, right? Because this codon attached to the anticodon of the tRNA. Complementary bases. Then according to the tRNA anticodon sequence, it carries a specific amino acid to the place. Then this amino acid are finally determined by the codon in the mRNA. Then what happened? As this process occurs, the phosphodiester, as this process occurs, these amino acids are getting connected. The bonds between the amino acids created by the ribosome. What is that bond? Peptide bond. Peptide bonds are creating between the amino acids. And then another tRNA carries another amino acid. and here. And the peptide bond is created between these two. This process continuously happens. As the peptide bond formed, this tRNA is detached from the mRNA as well as from the amino acid. When the peptide bond forms, the tRNA detached from the mRNA as from the amino acid and also it leaves from the ribosome. As the peptide bonds are forming, now tRNA leaves, ribosomes leaving, and finally it creates this amino acid sequence. Polypeptide sequence is created. So there is a particular amino acid pattern here. Amino acid pattern is here. This pattern is created according to this codon sequence. That codon sequence, according to the codon sequence, amino acid sequence is created. Amino acid sequence means this particular sequence will arrange according to a particular structure, which is called a protein. Now it is the character. It is the character. It may be keratin, it may be say multiple the protein, collagen, globin. So, because amino acid sequence determines the structure. Amino acid sequence determined by the codon sequence. Codon sequence was created according to the message present in the gene. Gene determines the codon sequence in the mRNA. According to the codon sequence, amino acid sequence is determined. Amino acid sequence, the polypeptide chains are created with the support of the ribosomes. tRNA is the one which carries the amino acids to the mRNA molecule and to the ribosome. This is called the translation process, which happens in the cytoplasm. That DNA determines our characters. The mRNA is synthesized and it comes to the cytoplasm. Now it's synthesized. The mRNA is synthesized.
Production of proteins. It needs to be processed. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, then see the entire process on time.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
See, this is the codon. This is the codon table. Now you can see when the codon is UUU. When the codon is UUU, the tRNA carries amino acid called phenylalanine. Or there may be a codon like UUC. So when the mRNA has UUU or UUC, the amino acid carried by tRNA is phenylalanine. And when the mRNA has a codon UUA or UUG, tRNA carries the amino acid called leucine. And you can see the red color one is given here. AUG. AUG is called the start codon of any the mRNA molecule synthesized in the human body. In not only humans actually. So that is in the eukaryotes. So when we consider the eukaryotes, the first amino acid present in each protein is methionine, because that is common. In prokaryotes, that is called the 4-mile methionine. 4-mile methionine the prokaryotes lage first amino acid again. Because all the mRNAs synthesizing AUG is the first codon, starting codon of any mRNA molecule. So therefore, the amino acids reached to that place is methionine. So when you compare all the proteins in the human body, the first amino acid is methionine. Why? The mRNA has the AUG as the start codon. The tRNA carries methionine for that. And also in the table, you can see there are 3 stop codons. UAA and UAG and UGA, stop codons. Stop codon means for the store, there is no tRNA to carry an amino acid to that particular place. So then from that onwards, the protein is not synthesized. I think tRNA is not carrying the amino acid to that. So in that way, so then when the mRNA has somewhere UAA codon, No tRNA carries amino acids. So then process stop there. Now methane up to this point only the protein synthesized. So when the protein synthesis need to be finished, that place has stop codon. In human and eukaryotes, we have 3 stop codons: UAA, UAG, and UGA. There. Okay, so then others are the relevant the codon sequences related to the amino acids there. So for example, when we take glycine amino acids, there are 4 codons maybe present in the mRNA. GGU or GGG or GGA or GGG. So whatever the codon sequence there, it takes glycine. tRNA takes glycine to the place. So this is the codon sequence presently.